Great. Thank you very much for asking me again to talk. So I've spoken at this meeting on and off over about 10 years, which is how long I've been working in FTD. And today I'm going to talk particularly about this project that I've been running for the last 3 years now, which is focused very much on genetic FTD. And so I might just start off with a step back and think about what causes dementia and what the pathway is. So this is my very, it's my simplistic way of thinking about it, because I'm a clinician and I do clinical research and I don't do much basic science research. So something kicks off your FTD. For some people, that is a genetic abnormality. For other people, it's something else that we don't really understand. But whatever that is that kicks off the disease, it leads through some biochemical pathways to an abnormal protein being deposited in your brain. And John has just shown you some pictures that we can now do looking at abnormal proteins deposited in the brain. So with those amyloid PET scans in Alzheimer's disease, and we can now start to look at tau, which is one of the important ones. Once that abnormal protein is sat in your brain cells, it causes you to lose brain cells, so you get brain atrophy. And once you've got brain atrophy and you're starting to lose cells, it alters all the level of the brain chemicals. And that loss of cells and the change in the level of your brain chemicals leads to the symptoms and signs of the disease. So in FTD, what is that? Well, the three major genes that we know cause FTD are these three genes that are called progranulin, tau, sometimes called MAPT, and this other one called C9orf72. And they, through a variety of pathways, lead to this deposition of these proteins, and the proteins that are important in FTD all have funny names, one called TDP43, one called tau, and one called FUS. And then, of course, you lose brain cells, and then some altered level of brain chemicals. We don't know much about this. We think sort of serotonin might be involved. And then, of course, it leads to these behavioral and language problems. And one of the key questions is, how long does this process take? So when this kicks off, so when these genetic and other risk factors start to cause the disease and then lead to this, how long is this process? So how long does it take to get to the symptoms and signs of the disease? Now, if you have genetic frontotemporal dementia, that means that we know that you come from a family, and it means that we can have a think more about this process, because we can look at people who don't have symptoms yet, but are people who may well carry the gene. So FTD runs in families probably in about a third of people. So in about 2/3 of people, they'll say, no one ever in my family. My parents died when they were old. No one in the family's had FTD. But for about a third of people, they will say, yes, there are people in the family that have had behavioral problems or language problems, and we can see that it runs in the family. The usual pattern, so there are lots of different patterns of how diseases run in families, but the most common pattern, and the one that's found in FTD, is this pattern called autosomal dominant. What that means is it runs from one generation to the next. So if you have genetic frontotemporal dementia, you have a 50% risk of passing it on to your child. So each of your children has a 50% risk of developing that problem. And it's variable by clinical diagnosis. So behavioral variant frontotemporal dementia is more genetic than the language problems. So we do see people who have the non-fluent aphasia who have genetic problems, but that's a lot less than a third. And we almost never see anyone with semantic dementia who has a problem that runs in the family. So it's mostly people with behavioral variant FTD. And we've been studying people for a long time. So when Martin Rohrer and John Hardy were here looking at for Alzheimer's disease genes, they recognized that lots of the families that they were seeing actually didn't have Alzheimer-like presentation. They noticed that quite a lot of them had a frontal-type presentation. And Penelope, who sat on the front row, spent a lot of her time traveling around and collecting people and families who had a variety of types of dementia, but particularly had an FTD-type dementia. And so what we've done over many years is start to study people who are in this pre-symptomatic period, or they are at risk of developing FTD. So that means that they are children of somebody who has genetic FTD, and that means they are at 50% risk of developing it. So some people actually opt to find out whether they are not just at risk, but whether they actually carry the gene or not. I won't ask you, usually in a professional audience, I will ask how many people would have that test. And actually, the reality is that virtually the vast majority of people opt not to know that they carry the gene. So in most studies, not just in frontotemporal dementia, when you have a disease that passes from one generation to the next, when you, as a child of somebody with genetic FTD, knows that you're at 50% risk, only about 10% of people opt to find out whether they carry the gene or not. So 90% of people will opt to stay not knowing whether they are somebody who carries the gene or not. So what we've done over many years is just follow all people, whether they know whether they've got the gene or not, knowing in our minds that 50% of those people are likely to go on to develop genetic FTD. And so going back to that thought about how long it is, that process of that disease process happening. Have a look at this scan. Some of you will have seen this scan before. What this scan shows is somebody who's came into our study with a progranulin mutation. So this is a cut through the head. You can just about see their ears. This is the left side. This is the right side. And what you can see, these are a number of MRIs made into a video taken 3 or 4 years in a row. So it's 3 or 4 MRI scans together. And what you can see over time, it's on a loop, is that this bit, the temporal lobe, and then just the bit next to it, the frontal lobe, are becoming smaller over time. Actually, the first scans are done before this person develops symptoms. So this is happening even before the development of any symptoms. So when we say that part of that pathway, thinking about loss of cells, of brain atrophy, from studies like this, we can say actually people are starting to lose cells even before they develop symptoms. So just going back to this about the genes involved in FTD, there are a whole lot of genes that have been said to be causes of FTD. But the 3 main ones, so these are the year in which they were discovered to be involved in FTD. The 3 main ones are these genes, tau, sometimes called MAPT, progranulin, and this one that's been discovered more recently, C9orf72. These other ones are much rarer. We almost never find people with these other gene problems. This particular problem, CHIMP2B, is seen in just a family in Denmark. So they're pretty rare, but it's these 3 main causes where we see people in our clinic. And so what we realize is that, of course, FTD is a relatively rare condition, and if genetic FTD is only a third of that, it's an even less common condition. So whilst we've been studying this for 15 or 20 years, it's very hard to really get a big study going and to really understand exactly what's going on in this pathway. And so 3 years ago, we contacted a number of research centres across Europe and Canada to try and get people together to try and study this in a bit more detail. And really, the key goal was about whether we could all work together and do the same assessment. So everybody does different things, and when we do research studies, traditionally people have always gone off and done their own study on a small number of people. But what we really wanted to do was try and get a mass of people together so that we could study this in a little bit more detail, which is just what you have to do when you have a relatively rare condition. So we decided to study people with these three main genetic types, progranulin, tau, and C9orf72. And we decided we'd study not just people with symptoms, but actually people in this pre-symptomatic period. So in this period where they are before they have symptoms, but they are a child or a sibling of somebody who knows that they have a mutation, so they know they are at 50% risk of developing it. And we have seen people, the youngest person in the study is 21. Their parents developed actually a progressive aphasia in their 50s, so 30 years different from the time when their parent developed it. But we're interested in seeing people really from 18 onwards of having an idea, because this is what will allow us to know what's happening in this pre-symptomatic period. And we did a whole variety of tests. Usually these take place over a single day, certainly here. You get a clinical exam, you get some memory psychology tests, you get an MRI scan, some blood tests, and we also, in people who agree, we do a lumbar puncture as well and take some spinal fluid. And so these are the centres that were involved in the first study. So James Rowe, if he's still here in the audience, from Cambridge, was the lead person in Cambridge. And so the two centres in England have been in London and Cambridge. We've got a couple of centres in Canada, and then Sweden, Rotterdam, lots of centres in Italy, and then over in Portugal as well. So these were quite a lot of the big European centres that see lots of people with genetic FTD. So back to this picture of thinking about what this is. So the question we want to know is what actually is going on in this picture. And so are there ways in which we can measure it? Well, we obviously can know whether someone's got a genetic problem. But how do we know about this abnormal protein deposited in the brain? Well, John Schott's just shown you the pictures of these PET scans. At the time when the study started, tau PET imaging wasn't around, so at the time we weren't able to do PET imaging to show this, but we think we may be able to see some of this in spinal fluid tests. And then this loss of brain cells, the brain atrophy, well, that's why we wanted to do the MRI scan, to have a think about whether we could see that changes. We don't have any good ways of measuring this, the altered brain chemicals at the moment. But obviously, there are ways of measuring symptoms and signs, doing psychology tests, and doing a clinical exam. And so we call these things biomarkers. So that means a marker of a biological process. And what we're trying to look for in this study is whether there are changes in biomarkers. So what this graph shows is that at some point, this marker, this biomarker, is normal. And then over time, it changes to become abnormal. And this is a sort of theoretical picture of what we think is going on. It's sort of the same picture as the one before. So we think that there are going to be markers in the spinal fluid and serum that change first. That's going to be measuring that change in the abnormal protein. And the same in the PET scans. We think that the PET scans are showing us change of the abnormal protein deposited. And then there are a variety of different MRI types that show us that we can see changes of brain atrophy. And then we see this change in the psychology tests. And then, of course, over time, people develop problems functionally and develop their behavioral and language symptoms. And what you'll see is that I've split this graph into what I would call clinical frontotemporal dementia, so the time when you have symptoms, and this pre-symptomatic period. And what we theorize is that actually a lot of these processes of the disease being triggered, of the protein being deposited in your brain, and of the cells being lost, is all happening in this lighter grey pre-symptomatic period. The question is, if this is time, what is this time period? Is this time period 5 years? Is it 10 years? Is it 20 years? We know that people have a genetic abnormality from birth. So the question is, at what point are we going to start seeing these abnormalities? And that's the key goal of GENFI. And so we have just had our very first paper published in one of the very big journals, Lancet Neurology. It's come out this month in Lancet Neurology, and I'm going to walk you through the changes that we found in our initial results. So we freeze our data every so often, so we say, right, we're going to stop all the data that's in at the moment. We'll just look at all the data up to this time. So that was at the end of 2013. And these are the sort of people that were in the study. So we'd recruited 220 people in total. And you see they're split. Most people are from progranulin families and less from the C9 or from the tau families. And we saw, say, although 220 people, actually, we see lots of people always from the same family. So only 76 families. And what we do is people come into the study, and most people will say, actually, look, I don't want to know whether I carry the gene or not. And we say, that's fine. What we do is one of our geneticists will anonymously test the sample, and then one of our statisticians will look at the results. So I don't know, none of the clinicians or any of the psychologists or anyone that's actually involved in seeing people will ever know whether they carry the gene or not. So you don't need to know whether you carry the gene or not to be in the study. And none of us who ever see people, it would be unethical for me to know if somebody themselves didn't know whether they carried the gene. It would definitely be very unethical for me to know that. And we're very clear with people that we as clinicians never find out whether they carry the gene or not, but it's important for the study to know who carries it and who doesn't. So that's done anonymously by a lab person and by a statistician that looks at the results. And so you see that it splits between people who don't carry the gene and people who do carry the gene but are in the pre-symptomatic period. So what we looked at, actually, it's very helpful. So instead of recruiting, most research studies have to recruit a control group, people who are an age-matched group of people who can match up with the people who are the people with the disease in the group. We don't need to do that because our controls are the people who don't carry the gene. So we have an automatic control group. And it's a really good control group because they've been brought up in the same environment. They are the same people. They are the siblings of the people who do carry the gene. So actually, we have this natural control group of the people who don't carry the gene. And what we saw in GENFI is that actually around 5 years before the predicted onset, and we don't know, of course, we don't know when people are going to get their symptoms. But we look at the mean age at onset within a family. So we look at all the people in the family that have had developed symptoms, and we say, right, we're going to say it's probably sometime around the mean age at onset in the family. So that's how we predict when people are going to get symptoms. And we see that, unsurprisingly, people get problems with frontal lobe function, and people also get problems with naming tests. And that happens, actually, we can see changes around 5 years before symptom onset. Very subtle changes, but with the large numbers of 220 people, it allows us to see those very subtle differences. And actually, what we see is that the onset for loss of cells, so the onset for atrophy, appears to be about 10 to 15 years before symptom onset. So very subtle changes in the brain, but nonetheless, we're seeing changes 10 to 15 years before people are predicted to develop symptoms. And I'll show you this in a series of graphs. Because what we looked at was not just whether the whole brain changes, but which bits of the brain change. And I'm not sure if people have talked about this today, but in thinking about where the different bits of the brain, there's obviously the frontal lobes and the temporal lobes. Those are obviously important bits. And then further back, there's things called the parietal lobes and the occipital lobes. But there are other sort of two bits of the brain. There's a very central bit called the cingulate, and there's a bit sort of deep hidden in called the insula. And the insula is sort of a bit that's right between the frontal and the temporal lobes. And interestingly, what we see is the first one to change is the insula. What this is showing on the bottom is time to expected onset. And what this is showing is the difference between people who carry the mutation and people who don't. So if there were no difference, of course, it would just carry on along the 0 line. But what you're seeing is that this starts to drop down and become increased loss of cells. So this is showing increased loss of cells over time in this particular part of the brain called the insula. And it becomes significantly different between 10 and 15 years. And the order in which things are affected are the insula, then the temporal lobe, and then the frontal lobe. Well, we know that. That's frontotemporal dementia. So frontal lobe, temporal lobe, and the insula, which is the bit in between the frontal and temporal lobes. And then, actually, the disease spreads through the brain into the parietal lobes, the cingulate, this bit in the middle, and lastly, the occipital. So that you see these changes starting in this very small bit, the insula, somewhere between 10 and 15 years before onset, in the frontal and temporal lobes around 10 years before onset, and then starting to spread through the brain as you get towards symptom onset. And actually, if you look at the individual genetic types, you see different bits of the brain in the different genetic types going first. So in tau, it's actually the temporal lobe that goes first. In progranulin, it's this bit of the brain called the insula. And in C9orf72, actually, it's a bit deep within the brain, a bit of the brain called the thalamus. So it's interesting that actually, although these all cause frontotemporal dementia, in fact, they have slightly different patterns of cell loss, of which bit of the brain is lost first. So we've moved on with GENFI, and by the end of 2014, we'd actually recruited 336 people, and a bit more C9orf72, so lots of progranulin, and not as many tau. And so we're just starting to look at this data a bit more and look at the other bits, look at the spinal fluid and the serum and the other types of MRI. And in fact, this month we're moving to a GENFI 2 because we presented our work at the International FTD conference in Vancouver last year, and we had lots of people very interested in it. And we had lots of people saying, we really want to be part of the group. We think this is the right way forward. And so actually, we're doubling in size. We're going from 13 to 26 centres across Europe and Canada. And actually, every centre in Europe and Canada that sees people with genetic FTD will now be involved in the study. So we hope to be able to see 600 people, and we hope to be able to see them over time. So not just at one time point, but be able to see them at 3 time points. We are very keen to have people involved who'd like to take part. So if anyone wants to come and talk to me afterwards, if they're from a family with a known mutation in progranulin, tau, or C9orf72, very happy to talk to you about the study. And as I said, you definitely don't need to know whether you carry the gene to be in the study. We're very keen to have people from 18 onwards from families of progranulin, tau, and C9orf72. And really, it's the only way that we're going to find out what's going on. So just to say the other centres, we are going to have, for those of you from further afield in the UK, both Manchester and hopefully Oxford are also going to join. So we'll have 4 centres within the UK, but we're also having Spain and France, Germany and Belgium also involved. So it's going to be a much larger study. We're hopefully going to get many more people involved. And the key questions come back to that diagram of what's going on, which is when is the best time to treat people. So ideally, if we had a drug that could stop this abnormal tau protein or the abnormal TDP43, when would we give it? So of course we'd give it to people at any stage, but actually when is the best time to give it? When are we going to be able to stop that brain cell loss? And the answer to that might be 20 years before people develop symptoms. It might be well before actually the time of when people develop symptoms. The second question is, how do we monitor whether the drugs are working or not? So we need to know what's happening over time. How quickly is tau being deposited? So on a tau PET scan, how quickly is that change happening? So that if I give a drug that stops tau from being deposited, how do I know whether that's working or not? So we need to know a little bit more of what's changing over time. And also, how many people are really needed for a trial? So we don't know the answer to that. Is it 20? Is it 40? Is it 80? So what we can look at is, well, if we know that your tau is being deposited this quickly, that allows us to work out, well, if our drug changes it this much, how many people are we actually going to need to be in the trial? And there are new trials on the way. So you're going to get a talk from Justin, who's standing at the back at the moment, who's going to talk to you about drug trials that are starting. When I first started talking at this group, there was no talk of doing drug trials. Just in the last few years, there's now finally talk of doing trials in people with FTD. The particular trial that Justin is going to focus on, I think, is a trial from Forum Pharmaceuticals, and that's specifically for people with a progranulin mutation. Unfortunately, so far they've said that you can only be in the study if you know that you carry the mutation, so they are going to include people both with who are symptomatic and also pre-symptomatic. But I shouldn't preempt Justin's talk, otherwise he'll have nothing to talk about. So, that's GENFI, and John overran a bit, so I'm going to catch up and only have 5 minutes so that you actually get to your lunch on time. So what I want to do in just the last 2 minutes is actually just quite step away from GENFI and just talk about a website that I've developed in collaboration with a number of people. So this website is called FTD Talk. It's at www.ftdtalk.org. And it's supposed to be a website for the whole FTD community. And I'll show you what's on there. It's actually an international website. So although I run it with 2 other people here at UCL, we have contributors, and I can see at least a couple of people in the audience who've contributed from across the UK, but also across the US and Australia. And we are planning to have translations of the site into other multiple European languages. The main idea is for us as researchers to be able to tell you in a simple way what's going on in FTD research, to keep you up to date with FTD research. So usually once every couple of weeks, when an interesting paper comes out, we write a very short 2 or 3 paragraph introduction to that bit of research. And we try and write it in a layperson way. Now, scientists aren't always great at writing in a layperson way, so we are doing our best. And if it seems complicated, then I try and edit it and make it a bit better, but we learn as we go along. So on the front page, what you'll see are these new, or these stories. So this was the one that was up earlier in the year, talking about inflammation, which is a thing that we're particularly interested in at the moment. But actually, there's a little bit about a sort of introduction to FTD. So if people have never heard of FTD, there's sort of a lay introduction of what it is, and there's a lay introduction to about the genetics and about the pathology and the imaging. So there's just a sort of introduction to that. And you can also get, for those of you a bit more interested and a bit more science bent, Jason Warren, myself, and Martin Rohrer wrote a review about frontotemporal dementia that was in the BMJ, the biggest British medical journal, in last year. And so you can download that for free as a PDF from the FTD Talk website. There's a bit of a glossary of terms. So we talk about all these funny terms. What do they all mean? So there's a sort of lay introduction to what all those words actually mean. What do we mean when we talk about tau and TDP43? What's a gene? What's DNA? All of those things are on the website. And lastly, there are actually some fact sheets about diseases. So there's some fact sheets about FTD in general, but about all the different subtypes, a bit about some other conditions, about treatments, and other things. So these are very small, short fact sheets that you can go and download that'll tell you a little bit about the disease. So we really hope that this website will be something that people can go to regularly to try and keep an update about FTD and about the research that's happening. We like people's feedback to tell us what they think and what they would like on the site. And we try and keep it sort of focused so that people will be able to. It's written in UK English, and I correct the US people when they send things to me. So behavioural is spelt correctly. But nonetheless, I do want it to be a universal audience. So we do have some things that are a bit more US focused sometimes, or Australian focused. And the website is here. For those of you that are actually, I should have put our Facebook site as well. So we have a Facebook site, FTD Talk, which you can look at, which people seem to refer to Twitter. But if you're on Twitter, then often we tweet about recent papers and research that's come out. What we try and do is at international conferences, one of us will live tweet from the conference and talk about important things that are happening there and then. So if someone talks about a new drug trial or someone talks about new work that's coming out, we try and live tweet from that conference about what's going on. There's a GENFI website as well, so genfi.org.uk. That is my email, but I'm happy to stay around to answer questions, or if people are interested in any of these things, or particularly in GENFI. Thank you very much.